So when we get some of the tumor samples from the clinics, so Tanya Heim, our research coordinator, will bring them over. We'll actually take them into our tissue culture room. We'll dissociate them, which means take the tumor apart and get it down into individual cells. And so those cells, we make a suspension of those and we can actually plate them both 2D on plastic, but we can also grow them in 3D, which is really exciting because when tumor cells are growing in your body, they're almost like an organ themselves. They have different cell types supporting them, and they also even cause sometimes blood vessels to reroute to them. So we can actually grow these in 3D, which more accurately represents how they grow in your body. Anytime you take a cell out of the human body, it is going to change a little bit. And we do see that, but by growing them in 3D, we can try to minimize those changes. And we've actually published results in our group specifically looking at sarcoma. So we compared it to growing 2D, like everybody's been doing for decades in science, and then also in these 3D spheroids. And we found that the ones grown in the 3D spheroids mimic exactly the expression profiles that we found in the original primary tumors, which is a really exciting finding. To be honest, I don't think there are very many sarcoma tumor banks in the country. I think we might have one of the largest here at UPMC, University of Pittsburgh. And right now we have over 45 different subtypes in our bank, which is absolutely incredible. Working in an environment where you study rare tumor types, it's incredibly hard to get cell lines and models to study your disease. So getting samples from patients, it's incredibly valuable because we can bring those back to the lab, we can establish them, we can make cell lines, we can make new tools to do different types of screening, figure out new therapeutic targets. And there's a couple things you want to look for when you're growing primary cell lines. So you want to make sure you don't have any contamination. So when we get the cells, the tumor sample, we actually treat them with various antibiotics and different washes to clean them so that if they picked up any bacteria on the transport or from coming out of the surgery, we can, hopefully eliminate that. Another thing is the outgrowth of cells that are not tumor cells. And so this is where it's really important to do something that we call immunohistochemistry. We use special stains to look at the cell types that we're growing, and we actually work with some board-certified pathologists at Shadyside Hospital, and they help us determine if the cells that we're growing are still like the original tumor type. So that's incredibly important to validate that before you move forward with all of your different assays. So individuals that would be interested in working with us and collaborating with us on this tumor tissue bank would be any kind of sarcoma researchers across the country or across the world. Dr. Weiss and I are always looking for new collaborators, and we're always excited to share these tumors because the patients gave these samples hoping that they would be able to help someone else in the future. And so we don't want to silo things just here at University of Pittsburgh. We want to work with everyone. When you work with such a rare tumor type, it's very important to reach out to others and try to work together to combine your resources.